My name is Ina Bando and I have been working in the SCRO lab. So this summer I researched cell migration in 3D microtissues and developed methods to accurately track cells on this platform. Cell migration is the key to many phenomena in life. For example, it plays a big role in embryonic development, tissue repair, cancer, inflammatory diseases, and even mental disabilities. Being able to track cells accurately can lead to advancements in topics related to my research, which can then help treat diseases, remodel tissues, and get closer to curing cancer. But cell tracking is particularly difficult on 3D microtissues, which is the platform my lab works with. Although these 3D tissues are great models for mammalian cells because they mimic an animal model without actually using animals, they also have multiple cell layers, which makes it difficult to track individual cells. There are differences between the layers in contrast and brightness, and because of these differences, analyzing images from 3D microtissues is much harder and more complicated than analyzing images from other platforms. So the goal of my research this summer was to address these difficulties by trying to make cell tracking on this platform easier. We took two approaches to tackle this problem. The first was to use molecular cloning techniques to create cells that express two fluorescent markers, and the second was to optimize a sequence of parameters in an imaging processing software called Cell Profiler. We were successfully able to create cells that express the fluorescent markers, red in the nucleus and green in the cytoplasm. To do this, we combined multiple DNA fragments and introduced the genetic material into mammalian cells. Then, we seeded these cells into a 3D microtissue and imaged them using fluorescent microscopy. We saw that the individual cells were more distinct because of the nucleus markers, which was what we had hoped to see. And lastly, I manipulated various parameters for these images in Cell Profiler and was able to optimize its ability to identify and track cells. A future goal with these processed images is to quantitatively measure how well a cell tracking method worked. Using these measurements will allow us to compare previous methods with new cell tracking methods. Our new method has the potential to initiate advancements in cell migration research, which in turn can positively impact medicine involved in wound healing and curing cancer and other diseases.